So we're going to pick right up where we left off from last class, and we're going to talk about the synapse. And so we talked about the electrical transmission of the action potential, which is the electrical impulse that happens within the neuron and propagates down the length of the axon, jumping from node to node of the node of Ranvier. And then we talked about the fact that this signal goes all the way down the length to the axon terminal. And then here we can see this would be basically an extension of this is the terminal button right here. Okay. And we're going to talk about how the chemical transfer occurs between neurons so that you can kind of see how this works. So this right here, this gap right here is called the synapse or the synaptic cleft or synaptic gap. Everything here is considered the presynaptic membrane, and that is essentially the sending cell. And so this is sending information to another cell. In this case, this could be a cell soma or a dendrite connection from one next. Now, one thing that I do want to mark here, make sure that you guys are aware, is this right here is a gap. The cells actually are not physically connected to one another. There is this little gap here where the chemicals will be transferred. So this is the presynaptic membrane, and here we have the postsynaptic membrane or receiving membrane. Now, this is the area that has specific receptors to receive the information, the chemicals from the previous cell. And these receptors, think of these as like a lock, and the chemicals themselves as the key. And so in the presynaptic area, there are vesicles, for example, or sacs, where there may contain chemicals or neurotransmitters. These can be synthesized in this axon terminal area, and can also be synthesized in the soma or cell body. It depends on what kind of chemical it is. And then if it's synthesized in the body, it's going to then travel down the microtubules into this area. Even if it's synthesized later on, parts can, like the vesicles, can actually be transmitted through those microtubules. And then that will then be released into the presynaptic cleft and then bind to those receptors that are on the postsynaptic membrane. And so this is going to be how we're going to talk about this sort of process occurring. So what I want to spend a little bit of time on is the synaptic vesicles and how this looks in terms of information and chemicals being synthesized and then transferred from one cell to the next. So vesicles, these are the small little sacs. They're rounded objects that are filled with chemicals. In this case, we're talking about neurotransmitters. And so it will fuse, the vesicles actually fuse with the presynaptic membrane in order to open up, to create an opening so that the chemicals can then spill into the synaptic cleft. So this right here, so you can see that this is one that's intact, okay, and it's like moving toward here, and then this one has actually fused with the presynaptic membrane to basically open to allow for that chemical to come out. And in order for that fusion to take place, you need to have the calcium channels, voltage gate calcium channels activated in order to allow that to happen. And we're going to see this occur right here. And so as you can see, we have basically a terminal button or a terminal bouton here. And this would be where, the synaptic cleft would be, because it would be attached to another neuron. And here you can see that this is a blow up, okay, of a vesicle that contains chemicals here. And so as you can see, this is sort of the process. We have a calcium channel here, Okay, and then you can see that it's actually fusing with the membrane. It allows it to open up, and then the pore is going to widen to allow for the release of all the chemicals in here, and they are going to then enter in to the synaptic cleft, and then the receiving cell will then take that information and be able to determine what chemicals it's going to receive. So once we have the chemicals, the neurotransmitters, released into the synaptic cleft, what is the process that happens after that? Well, in this case, what we see is that receptors need to be activated on that receiving end, the postsynaptic membrane. Depending on the kind of chemical it is, it's going to open up ion channels for that specific neurotransmitter dependent channel. It can activate a postsynaptic cell, it can inhibit, it can mimic other chemicals, lots of things that can happen here. We're going to talk about the basics, and then we'll get into some really good detail later on. So here, another interesting thing that happens. So here we have the terminal button, and then here is, let's say, a dendrite of some other cell. We have the synaptic cleft right here. So there are receptors here, of course, to receive the chemicals that are coming out. But what's also really interesting is the presynaptic membrane also contains receptors. And so these receptors' jobs are to make sure that everybody's doing their job and that the level of neurotransmitters, the numbers of neurotransmitters that are being released are at the correct level. And so if, for example, this cell is releasing too many neurotransmitters, this sort of autoreceptor here will then disengage and say, uh-oh, we have a problem here. We need to shut off the release. And so it's a mechanism to allow for the cell to determine if there's too much information being released, too much chemicals being released, or not enough. So it's going to regulate that process. So we have receptors on the postsynaptic side, which is it's receiving, and we have receptors on the presynaptic side, which is the sending. So those are both going to give information to and from the cell to determine what we have that needs to be sent and received. Now, there are two types of receptors that we're going to talk about. Their differences lie in how quickly and how quickly the binding happens and how many we have. So there are ionotropic receptors and metabotropic. And so we're going to first talk about ionotropic receptors. And so what we have here is these are about 20% or so of our receptor types, and it contains a binding site for a neurotransmitter and will actually open directly an ion channel, which is why they're called ionotropic. So here we can see that we have a binding site for a receptor. Here a neurotransmitter chemical is going to bind to that site and open up the channel to allow for a specific ion to get through. And so this is a much faster type of connection and allows a much faster change in charge in the cell. So we only have a few of these, so about 20% or so of the receptors are ionotropic. And the idea is that these are the purpose of these is to have for really fast transmission of ionic molecules. So looking at metabotropic receptors, this is about 80% of our receptor types. These actually do not directly control ion channels. It's not as fast, okay, when looking at the kinds of movements of chemicals and ions. So here would be an example of a metabotropic receptor getting a binding, having a neurotransmitter binding to the metabotropic receptor. It's going to activate this G protein, which is here, and then that's got to send a signal to break away an alpha subunit. And then in this case, and not until step 4, do we see that ions are starting to enter the cell. And then, here you can see in this case, just more steps in the process. Okay. Here we have a neurotransmitter binding to a receptor, activating the G protein again. And then here the alpha unit is going to break away, and then we have an enzyme that's taking control, giving information to the second method. There's a lot of things going on here. I'm not going to expect you to memorize this process, but what the point is that there's all these extra steps that are occurring that require this metabotropic receptor to activate in order to open a channel to allow for the ions to move in and enter the cell. So here we have this process. It's very quick. Okay, basically we have a binding and then the channel opens. Ionotropic is very fast, very directly affects the ion channel. Here metabotropic, and these are two different sort of processes here. It requires additional messengers. It requires a different activation of proteins and enzymes to allow for that channel to open, for the ions to move across the membrane. And so, we only have a few of the ionotropic, but the metabotropic this is how more of the receptors work. So looking at the postsynaptic potential, and this is where we're going to talk a little bit about how the messages get across to determine if the cell is going to fire or not. So when we talk about remember we talked about the resting potential, we talked about the action potential. Well, the postsynaptic area also has a potential, and so we can have an excitatory potential, or we can have an inhibitory potential. And so remember that when we have this kind of this graph here where we have the -70 millivolts, that's the resting potential, and then we hit the threshold of -60. So we're here, and then we get this stimulation, and then we get boom, once we hit the threshold, we get all the way up to about +30 millivolts is the peak, and then we see this going down here. So we have this being depolarization. Okay, and then we see drop my pencil. Down here we see this repolarization. Okay, and then we see here this dip down, which we call hyperpolarization. Okay, and so the hyperpolarization is what we refer to as sort of inhibitory, where we don't want something to fire. Excitatory would be depolarizing. We want, in the case of firing, you want this process to make a cell more positive in terms of charge. And whether it's going to be an excitatory postsynaptic potential or inhibitory postsynaptic potential will be determined by what ion channels are being opened. This is different than an action potential. Okay, so I don't want to confuse you that the process. This isn't necessarily the process. I want to remind you what depolarization is and hyperpolarization. Okay, remember the process. So depolarizing is making that that cell more positive, whereas hyperpolarization is making it essentially more negative, right? But what this postsynaptic potential is essentially determining what the environment, if it's going to be more likely to be excitatory, depolarize the message or the neuron, or it's going to be more likely to hyperpolarize or inhibit it. And what happens is that these will add up, okay? So they'll add up to determine is this going to lead to an action potential? So these IPSPs and EPSPs, these are not action potentials themselves. These determine if it's going to add up to an action potential or not, okay? And remember, if it has an action potential, if you have an action potential, it's an all or none. It's not a partial. Once the triggering has occurred, the cell will take over and then initiate the rest of the action potential. So these postsynaptic potentials, they are changing the local membrane potential around a receptor to determine if enough stimulation is going to occur for the action potential. Okay, and so this is kind of the description of how a postsynaptic potential would work in opening the channel to determine if it's going to actually fire or not. And it requires, of course, the activity of the ion, okay, the sodium, okay, and if there's an influx of sodium, it's going to result in a depolarization, excitatory, more likely to be an action potential. Okay. In the case of potassium leaving the cell, right, an efflux or removal of potassium, that is going to cause the cell to experience a hyperpolarization or be more likely to be more negative. So that's going to inhibit the process and be less likely to engage in an action potential. Okay. If we have an influx of chloride entering the cell because that's more negative and making this environment more negative, that's going to again result in a hyperpolarization, inhibitory, and not result in a firing. If we have calcium channels opening up and binding to enzymes, okay, that's going to impact the likelihood of of the cell becoming more positive and be more likely to depolarize. Okay, so here's another way to look at it. Okay, so if we have all these binding sites, and in this case, red would be excitatory or an excitatory postsynaptic potential. The more ions that are going to result in a closer positive charge, that is going to be more likely to produce an action potential, because all of these EPSPs are going to add up. And that information is going to be more likely to make this neuron engage in an action potential and become excitatory. If you have connections that are closer to the axon hillock, okay, then those connections tend to be granted a little bit more power and be more likely to be EPSPs and result in an excitatory reaction, which would be a depolarization, which would be an action potential. So not only are we seeing these things add up, okay, but if also we see connections directly to the axon hillock, we see more likely to be an EPSP. Now, in the case where we have both EPSPs, okay, so both excitatory message areas here that are sending the signal of making this area of the neuron more positive, resulting in a depolarization, okay. Now we have on this side, we have more inhibitory messages coming through and saying basically, no, don't fire, okay. And so what these channels are opening and allowing for negative ions to enter in, resulting in a hyperpolarized message, or IPSP, which is being inhibitory. If these add up enough to counteract the EPSPs, the end result is no action potential. If you don't have enough of these IPSPs adding up to inhibit that message, then what's going to end up happening is then what will overpower is the depolarization, and it's going to end up being so positive it's going to cross that threshold and result in an action potential. So it just depends on where these connections are. Okay. So like, are they on dendrites? Are they on cell bodies? Are they on axon hillock? How are they adding up? Are the IPSPs overpowering the EPSPs? And whatever overpowers and results in a difference in charge that matches the threshold will determine if this cell is going to fire or not. So another interesting thing that can happen is is a couple of mechanisms that can happen at the postsynaptic area that can essentially terminate the postsynaptic potential. So depending on what chemicals are being sent through, we can have reuptake happen, where essentially those receptors are taking back in the chemicals that they're releasing. And so some of you might be familiar with SSRIs. Okay, so SSRIs is a selective serotonin reuptake inhibitor. So when you take an SSRI, it's actually cutting off the reuptake receptors and is not allowing the process of the cell to take back in, to turn that mechanism off. And so because reuptake is being inhibited, it's actually creating more neurotransmitter to be available in the synaptic cleft. And so it's causing the postsynaptic side to take in more neurotransmitter. So that is an example of the reuptake process, okay? But you can actually see that the reuptake process can also work to sort of terminate the potential at the postsynaptic level. So these are two different processes, but I'm bringing in the SSRI to describe the process of reuptake. Okay, so it's basically taking back in some of that neurotransmitter that it's sending out. Okay, another mechanism by which the postsynaptic potential can be terminated is through enzymatic deactivation. So essentially, when it's the ion is being sort of attached to an enzyme, it's going to terminate that process and just not let it continue on in terms of sending the message through. So again, these are just a couple of the processes that occur. Final thing that I want to talk about, again, this idea of the receptors being on the pre versus the post synapse area. Okay, autoreceptors. So if we have here, for example, okay, this is the terminal button, and then this is the synaptic cleft, and then this is the sending end, and this is the receiving end here. Okay, autoreceptors that are on the presynaptic membrane, these are available here. Okay, these autoreceptors are monitoring the release of the neurotransmitters. So let's say we have this process where this autoreceptor is receiving back what it's given. So here it's releasing the neurotransmitter, and the postsynaptic cell is no longer taking as much of that chemical in. The autoreceptor will then be monitoring this. And the autoreceptors that are on this presynaptic membrane will adjust the release of the chemicals. And so what it can actually result in the termination of the postsynaptic potential, because it says, look, stop, let's inhibit this process. We don't want the release of more chemical because it's not being received. Okay, and so you don't want these chemicals to just, have to basically dissipate, which is something that they do, but it being taken back up into that autoreceptor will allow for this particular sending cell to monitor the release of chemicals to determine, okay, what are we going to do with the rest of this neurotransmitter, this chemical? So for example, dopamine, the autoreceptor says, hey, stop releasing dopamine because we're wasting it. Okay. So that's just kind of an overview of the things that we see in terms of receptors and sort of the end of the action potential and that the chemical release with that. So if you have any questions, you can email me, and make sure that you read the chapter material along with this because it helps supplement some of the things that I covered, and that should help you sort of self-study this part right here.